Hello, good morning, everyone. I hope all of you are doing well this morning. So we will begin right now. As I mentioned last time, I assume you have watched the lecture recordings, and we are now going to go through the study guide. And you might have attempted this on your own already. And if you haven't already, that's okay as well. We'll be going through some of the questions together. And if you haven't had a chance to watch the recordings yet, you might want to still watch it afterwards. Okay, you might not be able to follow 100% of everything I go through, but do stay, and you can catch up afterwards. All right. So here is the first question. List 7 characteristics of living things. Okay, so we're just gonna write some of them down. I'm doing it in no particular order, okay? I'm just gonna write down as I remember them. So for example, one of the characteristics of all living things is that they exhibit order. Okay, so for example, they are complex. There's some complexity to it, right? There is symmetry, right? Some kind of organization, right? And so on and so forth. So there are these built-in orderness that living things exhibit. Another one is the ability to maintain homeostasis. Can somebody tell me, either you can unmute or type in the chat box, what is the meaning of homeostasis? What does homeostasis mean? Anybody? That's right. So a few people say internal balance, right? So the homeostasis means you want to maintain some kind of equilibrium in the body, okay? Maintain an internal balance. So for example, we as humans, we like to maintain a body temperature of 37 degrees Celsius, okay? That is homeostasis, okay? If it's too cold, you shiver, and you would generate more heat to keep yourself warm. And if it's too hot, you would sweat, and the sweat would help cool you off. So that's one example of homeostasis. Okay, so it's a type of self-regulation, exactly, right? So and in class, right, in the recording, I said homeostasis can come in one of two forms, right? They could be physiological, okay, or they could be behavioral, right? So physiological means that you are going to, your body is going to do it for you. Okay, so like the sweating, like the shivering. Okay, behavioral is something that you deliberately choose to do. Right, so if it's too cold, you put on a jacket. That would be a type of behavioral a mechanism for maintaining homeostasis. Okay, so another characteristic of living things is they grow. There's going to be growth and development. Okay, so we look very different now than we were when we were born, because we grew a lot, and we also develop. And in some other animals, the juvenile form looks completely different than the full-grown adult form, right? Think of like a butterfly. The caterpillar looks very different from the actual butterfly. So all living organisms will exhibit some form of growth and development. Although it's a little bit subtle for some species, if you think of like a bacteria, they do grow, but then not in a sense that we grow, right? Like you wouldn't be able to tell just by looking at a bacteria that's like, freshly formed compared to a week later, right? Or even like 2 weeks, 3 weeks later, right? It's pretty much going to look the same on a cellular level. So sometimes the growth is not as evident for some living organism. Another characteristic is all living organisms, they are going to require energy. So we humans, we consume food to produce energy. If it's we're talking about like a plant, for example, then they are able to extract the energy from the sun and make their own food. Okay, but regardless of the method of energy acquisition, all living things are going to require energy. Another one is that they respond to environmental stimuli. Okay, so a stimuli, which is the plural for stimulus, US, right? This is basically some kind of changes, right? Changes in the surrounding. Okay, so that change might prompt a series of physiological or behavioral response such that the organism is able to maintain homeostasis, like a drop in temperature, right? Your body will cope with it. But it doesn't have to elicit homeostasis necessarily. If an organism senses danger, for example, it runs away. That's an example of response to environmental stimulus as well. If you think of a plant, sometimes some sort of plants, if you touch it, then they will close up, right? So that's a response to stimuli, right? Another example of response to environmental stimuli is plants tend to grow towards the light. If you have any house plants, you might see them bend towards the window. That's an example of response to environmental stimulus. It doesn't have to be like an immediate response. It could be like a very slow response over time. Okay, think of the plants again, right? It's going to take maybe a week or two before you see it bending towards the light. And if you shift the plant, it might bend the other way. Like the roots tend to grow towards gravity. Right? That's an example of response to environmental stimulus. Whereas if you think of like a human, right? Like if you shine the light at the pupil, right? You've probably seen this on TV. If you shine the light on pupil, you can see the pupil constrict, right? That's a very immediate response to environmental stimuli. Okay, but again, it's not about how quickly it responds. It's just the fact that living things will respond to the environment. Okay. Another one is reproduction. Okay, that one is self-explanatory. And what is the last one? Anybody remember what the last one is? The last characteristics that we talked about. Very good. Okay, thank you for typing the answers in the chat. They are able to adapt and evolve. Okay, so just to clarify a little bit here, evolve. I'm going to put a star. I'm going to come over here and say population evolve, not individual. Okay. So there is a good question. Is homeostasis and adaptation the same thing? Not really. Okay. So homeostasis happens on an individual level. I am able to maintain an internal balance. Okay. So again, if I'm cold, I shiver and then I warm up. That's something that an individual organism would be able to do. That's what homeostasis is. Adaptation is talking about features that the population has over time as a result of evolution. Okay, so how does that work? So let me give you an example. Okay, let's just say we have this imaginary organism. Okay, so That's the organism. Okay, let's give them some legs. And in this organism, okay, there are two variations of them. Okay, some has a lot of fur. Okay, others don't. Okay, and this fur is genetically inheritable. Okay, so whether they have fur or not is encoded within their DNA. Okay, so let's say the temperature drops to -70. Okay, for a couple years. Okay, for whatever reason. Okay, the climate becomes very, very cold. So these ones that does not have fur will tend to die off, right? And the ones that have furs, they would be able to survive and mate and have more offsprings than the furless one. Okay, so over time, right, you will see that the population will have more of these furry stuff. Okay, so then now we say the population is more adapted to the cold environment. Okay, how did they become more adapted? Well, the population has evolved, right? Basically, what that means is those individuals with a favorable trait in a particular environment is able to survive better, is able to have more offspring, passing on that favorable trait. So over time, the population will consist of more of those individuals than initial. Okay, so that is what evolution is. It causes the population to become more adapted. Now, it's important to know, again, right, the entire population evolves, not individuals, right? A misconception is this, right? It's thinking there are these, furless little things, and then it got cold. So what happens is they evolve and they start to have furs, okay? That's not what evolution means, okay? For the population to evolve, that particular trait would have already need to be in the population in the first place. Does that make sense? Anybody have any questions so far? Again, just feel free to jump in if you have any questions at any time. Unmute, type it in. It's all good. Okay. I want this to be an interactive experience. All right, moving on. We talked about induction and deduction. Okay, inductive reasoning versus deductive reasoning. Okay, so how are they different? What is the definition? What are some examples? So let's go through induction first. Okay, so induction, basically, you can think of starting with a lot of individual observations, and you take all these observations and you come up with one generalized statement. Okay, so from specific examples to a general blanket statement. Okay, so derive. Okay, derive just means come up with. Derive a general statement based on many specific examples. Okay, or we can say observations. Okay, so if I have to draw like a picture, right, you would be like, okay, 1 example, 2 examples, 3 examples, 4 examples, many examples. Okay, and then you're gonna take all these examples, and then you're gonna come up with a general statement. Okay, so you start here with these observations, examples, and you come up with a general statement. Okay, so an example I can give you is this. Okay. So you're like, fish is made up of cells. Okay, birds are made up of cells. And then what else? Like trees. Trees are made up of cells. Okay, and then I don't know. I'm running out of organisms. Bacteria. Bacteria are made up of cells. Okay, so all these things are made up of cells, individuals' observations, and then you come up with a general statement. Okay, general statement saying all living things are made up of cells. Okay, does that make sense? Induction and inductive reasoning, specific to general. Deductive reasoning or deduction is the other way around. The other way around. Okay. The reasoning. If you cannot read my writing, please let me know. I'll try to write better. Flows from general to specific cases. Okay, so you start with general statement. And from this general statement, this premise that you have created, you are going to apply it to different specific examples. Okay. So using the same example here, I will start with the statement, all living things is made up of, are made up of cells. Okay. And then let's say we discover the new organism that we were talking about. So this thing, right, from our evolution example, this ball thingy is a living thing. Okay. So based on this general statement, right, we can make a prediction, right? Ball thingy is a living thing. It is made up of cells. Okay, and then you can go confirm the prediction afterwards, right? Okay, so going from general statement to specific. Okay. Just give me 1 second. My daughter needs a little bit of help. 1 second, I'll be right back. Sorry. Hopefully they'll be back to in-class learning next Monday. But yes, okay, that's the difference between induction and deduction. Any question before we continue? This tends to be confusing for some people. Okay, well, let's show me that you actually understand. Okay, here is a poll question for you to try. Okay, so this could be one of those harder questions on the quiz. So go ahead, make a choice. Remember to read all of them first before making a decision. Will this lecture be on homepage so you can write the notes? What do you mean, Ashana? You mean you mean like where you can find this thing? Is that what you mean? Okay. I will tell you in a bit. I'm just gonna finish the poll question first. Okay. It has almost been 2 minutes, so I'm gonna stop the poll. Well, I guess some of you are still trying, right? So I will end the poll first. I'll share the results. And let me tell you where to find the stuff in case some of you is asking the same question. So if you go to the course homepage, right here, there, you can click on the scientific method study guide and you will be able to download a copy. Okay. If you don't have it now, maybe just write it down in the notebook and that would be okay as well. All right, let me go back to here. All right, so I can see the most popular answer is C. Okay, and then this is the second most popular answer. Let's go through it. Okay, so again, deductive reasoning. Right, start from general statement to specific. Okay, so let's take a look at the first one. All virus requires a host to replicate. Okay, so that right here, that is a general statement. Okay, that is a general statement. So that's looking good. Okay, and then COVID-19 is a virus. It must require a host to replicate. Okay, so that sounds reasonable. Okay, that sounds like what we're looking for. But let's read the other ones. COVID-19, Ebola, influenza, or viruses that requires a host to replicate. So this looks like a bunch of specifics, right? Okay, so that's same as saying, you know, fish is made of cells, trees are made of cells, bacteria is made of cells, right? So that's not what we are looking for. That would be an example of inductive reasoning, right? And then the next one, all viruses require a host to replicate. Okay, so again, that is a general statement. And then COVID-19 requires a host to replicate, therefore COVID-19 is a virus. So that also sounds, maybe that's the answer. Okay, we'll come back to it. And the next one, COVID-19 is a virus and it requires a host to replicate. Ebola is also a virus, therefore it also requires a host to replicate. So these are just like two specific examples. There is no going from general to specific or specific to general. It's just two separate examples. So that's not the answer. Okay, so now we are going to have to debate whether the correct answer is A or C. So at first glance, both seems to be examples of deductive reasoning because you're going from a general statement to a specific example. But let's examine the C here a little bit closer. Okay, so it says all virus requires a host to replicate. COVID-19 requires a host to replicate. Therefore, COVID-19 is a virus. So the problem with this one is that the logic is not quite correct. It might not be obvious when I use words like virus or replicate and then host, but it will become apparent what the problem is if I change the wording a little bit. For example, instead of virus, okay, I can say, let's say all birds. Okay, I'm going to change virus to birds. Okay, so all birds, instead of requires a host to replicate, I'll say all birds have wings. Okay. And then here it says COVID-19 requires a host to replicate. So instead of COVID-19, so I'm going to give another example, right? Instead of virus, I'm going to say bees. Okay. So remember, I'm changing requires a host to replicate to have wings, right? So bees have wings. So according to this logic, what would be the last sentence? Okay, birds have wings. All birds have wings. Bees have wings. Therefore, what? Does everybody understand my logic? Yeah, therefore, bees are birds, right? Bees are birds, right? So if you change if you change the example here to something that we can relate, then you see that the logic is incorrect. Even though you're trying to go from like a general statement to a specific statement, but then the logic is not correct. The reasoning is not correct. For that reason, this is also not the answer. Okay, so for it to be deductive reasoning, it has to be this way, right? All viruses requires a host to replicate. COVID-19 is a virus. It must require a host to replicate. Okay, so using the bee and bird example, it would be like all birds have wings, and then something is a bird, therefore it must have wings, right? Does that make sense? Please ask for clarification if you require any. Okay, so sometimes it's a little bit tricky like that on the quiz, right? You really got to take the time and read the question carefully. But don't worry, not all the questions are going to be this involved. Some are just going to be straightforward, and we'll do some of those later on. Okay, we are going to move on to the next part. List the steps in scientific process. Okay, so in scientific process, we begin with observation. Okay, so you're gonna make an observation. You're gonna be looking at things, gathering examples. Okay, and then you're gonna ask some questions about these observations that you're making. Okay, questions. Okay, why are things the way they are? Okay, why is it this? Why is it that? And then you can try to answer these questions yourself. You can try to make people like to call it like an educated guess, right? So that is a hypothesis. Okay, I don't really like to call it an educated guess. It's more like a tentative answer. Tentative answer. Okay, so based on your understanding, based on maybe previous experience, you come up with a plausible explanation, a plausible answer to the questions that you are asking. Now, in order for it to be a valid hypothesis, okay, it must be two things, right? You probably remember this by now. Must be testable, and that means you must be able to do an experiment to test out whether you're correct or not, and it must be potentially wrong. Okay, so falsifiable. Falsifiable. Okay, potentially wrong. Potentially wrong. Okay, so once you have that hypothesis, you're gonna predict the result. Prediction. Okay, if I'm gonna do an experiment, what would it look like if my hypothesis is correct? What would the result look like if my hypothesis is not correct? Okay, so you make a prediction of what the outcomes are. And then you are going to actually go on and do the experiment. And there are various things you have to consider when you set up the experiment. And this is actually important for when we do the beetroot lab. Okay, a lot of questions is related to what we're talking about, how to set up the experiment, how to make sure that things are consistent and the results are believable. And afterwards, you will be able to make a conclusion, right? Was my hypothesis correct? Was it incorrect? Okay, and if it's not correct, maybe I need to do another experiment. Okay, maybe I need to change my hypothesis for further investigation. So number 4, give an example of a non-testable hypothesis. Anybody want to tell me an example of a non-testable hypothesis? Okay. You can make up your own, or you can use one that you remember from the lecture notes. Probably better if you can make up your own. God created all living things. Yeah, sure. Okay, so we can do that. God created all living things. Okay, now remember, this is important, right? We are not trying to say whether this statement is correct or not. Okay, we are simply saying this is not something that's testable. Okay. A dragon can make fire. Sure. Love these. A dragon can make fire. Okay. And then there is other good one. The universe grows at varying speed. Yeah. I like that one. But you know, actually, that's interesting because a lot of stuff in astronomy is not testable. A lot of it is inferences made based on observations. But you know, we're not gonna go. We're not gonna go there. Let me see. Okay, there is another good one that I saw earlier. Where is it? Which is better, yellow or green? Yeah, I'm gonna say yellow. Yellow is better than green. Okay, so this is an opinion. You cannot test it. These are this is like what do you call it? Like fictitious stuff. Cannot test. This is personal belief. Okay, cannot test personal belief either. Now, very important though. Very important. I need to write down here because a lot of people make these mistakes. The hypothesis should not be a question. Okay, hypothesis here. Hypothesis, not a question. Okay, so we shouldn't say, you know, is yellow better than green, right? That would be the question. Okay, but the hypothesis is the tentative answer, right? Okay, so when you do the lab on the Beetroot lab later, and I ask you to write a hypothesis, do not phrase it as a question. Okay, a hypothesis should be a statement. Should be a statement. Okay, wonderful. Thank you for all the responses. Next one, give an example of non-falsifiable hypothesis. Okay, this was a little bit trickier to come up with. Anybody have some examples? Non-falsifiable hypothesis. Okay, so could be religion based. Yeah, everyone has a soul. Yeah, that one is more like not testable than non-falsifiable. All strawberries are sweet. Yeah, that is a subjective thing. You can't really say they're wrong. Okay, so I see a few other ones. Chocolate is better than vanilla. Again, that's not testable and also not falsifiable. Okay, so here is one that I can give you as well. Okay, this is something my parents like to tell me a lot when I was growing up. Okay, if you work hard enough, then you will get A+ in whatever. Okay, biology. Okay, for me it was English. Okay, if you work hard enough, then you will get A+ in English. But I never got that A+, so I told my parents, I worked really hard, but I didn't get the A+. And their response would always be, that's because you did not work hard enough. Okay, so you see the contradiction in this statement. The person who came with these statements can always come back and say, you didn't work hard enough. That's why you didn't get the desired outcome. Okay, and if you keep on trying and it still doesn't get it, then the counter argument is always, that's because you didn't work hard enough, right? Okay, so a non-falsifiable statement is one that can never be argued against. Okay, because they can always say that you know, you didn't work hard enough. Okay, you can always work harder. You can always work harder. So that's an example of that. Okay, seedless fruit are sour. you guys have very good examples. Okay, so that is it. Moving on. Give 2 examples of primary sources and 2 examples of secondary sources. So primary sources are like, you know, sort of like the first-hand experience, firsthand recollection of the situation. And an example of primary sources would be things like peer review, articles. Okay, so in science, if a scientist do some research and then they want to publish it, then they have to submit their findings to a committee, other experts in the field, and they would look at it and make sure that the data is sound, and then they would publish it. So that would be an example of peer review articles, and that is a primary source because it's written by the person who conducted the experiment. Right? Another example of primary sources, yeah, some of you said research journal, researcher or original findings. That's correct. What's another primary sources? Well, another one could be, could be diaries. Yeah, a personal recount of what happened on a specific day, for example. I'm thinking about like a journalist reporting live from a scene. Okay, so the journalist, the person is telling you exactly what's happening at the scene, right? Based on what they are observing. It could be a video, it could be a photograph capturing a particular event. That's another example of primary sources. Someone say textbook. Now, textbook is a secondary source, okay? Because the textbook is a collection of knowledge, right? That are discovered previously. For example, if you think of like a biology textbook, all the stuff in the bio textbook, the author did not discover them, right? It's discovery that has been made over centuries by many scientists, and it's just like a collection, a pool of intellectual material. So that would be an example of a secondary source. Another example would be textbook and encyclopedia, right? No one really use encyclopedia anymore. The only one people know is the Wikipedia, right? That is also secondary sources. News reports could be secondary if the person is not reporting at the specific scene. If they are just taking information gathered by someone else and then reporting it back to you, that would make it a secondary source. Yeah, newspaper, definitely newspaper, right? But the newspaper could also be primary source, right? If the person who's writing the article is at the location, right? Another one is like blogs, right? So all these are secondary sources. Okay, good. Let's do another poll. Right here, that is a simple one, I hope. Go ahead. Okay, and a minute is up. Yeah, very good. Most of you got it. Hypothesis, right? We talked about it, a tentative answer to your question. And how about this one? So these are examples of like easier questions on the test, the quiz, right, that you should be able to just hopefully answer without thinking much. Okay, and I will end the poll. And again, most of you got it. That would be an experiment. Okay. Let's go back to our lecture. Okay. This part here, I already did fill it out on the lecture recording. So if you haven't already watched it, you can go watch it and fill it in later. I'm not gonna go through this again, but I will write out the definition for independent variable and dependent variable for you once again, just in case you need a refresher. Okay, so what is an independent variable? An independent variable is the variable. First of all, a variable is something that can change, right? As opposed to a constant. Constant does not change. Variable will change. So an independent variable is the variable that is being changed or manipulated by the researcher on purpose. Okay, so that's the one that you, as a researcher, as the person who is conducting the experiment, you deliberately change that variable. Okay, and by changing this independent variable, you want to observe what is the effect, if any, on the dependent variable. Okay, so let me write it down for you. The dependent variable is the variable that is observed or measured by the researcher. Okay, to see whether or not changes in the independent variable would affect it. Okay, so by changing the independent variable deliberately, you want to observe or measure if there are any effects on the dependent variable. Okay, so for example, if I want to do an experiment to see whether or not having these live tutorial will improve students' performance. Okay, so I would have two classes. Okay, one class I would have live tutorial. One class I will not. Okay, so having live tutorial or not having live tutorial, that would be the independent variable. And then at the end of the semester, I would look at the grades of the two classes, right? The class with tutorial, without, and I would compare them. Okay, so the grades of the class average, that would be the dependent variable. And then I will be able to make a conclusion, right? Thus, changing whether or not there is live tutorial or not have an effect on students' grades. Is that okay? So here would be a bunch of examples, a bunch of scenarios, and you're supposed to find out which one is independent and which one is dependent. Again, this would be in the recordings. I believe it's the second one, the second one out of the three, and you can go watch it and fill it in. I didn't have the pen at the moment when I was filling this in the recording, so the writing was a little bit all over the place. So just keep that in mind when you're watching. Moving on. Answer each of the following questions related to Rady and Pasteur's experiment on the theory of spontaneous generation. Okay, so first let's recap what is the theory of spontaneous generation. To keep it, as simple as possible, spontaneous generation hypothesis basically states that living things, okay, arose or came from non-living things. Non-living things. Okay, so we already defined what living things are, right? It's the things with those 7 characteristics that we talked about. So people believed in the past that living things came from non-living things. Okay, and the reason is because they would see like what was it, like fish rain from the sky, for example, and so they'll be like, oh, okay, the sky, which is non-living, allows the fish to be spawned. Okay, so living things came from non-living things. Actually, it happened, I think last week. I saw a news feed that says, I forgot where, but if you search up like fish raining from the sky, you would definitely see an article last week that talks about it happening. It's like some kind of crazy weather, right? Scooping up the fish from nearby ponds and lakes and then sending them inland. Okay, so that is basically what spontaneous generation is. So Francesco Redi, he wanted to examine this theory. He wanted to prove it that it is not true. Okay, so what observation was Redi's hypothesis based on? Okay, so his hypothesis is basically that when a piece of meat is left outside, flies and maggots will appear afterwards. Okay. So the piece of meat, which is non-living, afterwards, you would have the flies and the maggots, which are living. They would appear afterwards. Okay, so Redi observed that, and then his hypothesis is basically, well, if the theory of spontaneous generation is true, then flies and maggots would be able to spontaneous appear from a piece of meat. Okay, so how does he go on and do this experiment? Well, in his experiment, he had 3 groups. Okay, so there are going to be 3 groups. Okay, there's going to be the first group, which is just an open jar, and then he's going to have a piece of meat inside. Okay, and then the second one is going to be a jar with net covering. Okay, so it's going to put like a little mesh here and dump a piece of meat inside. Okay, and then the last one, he's going to have a sealed jar. If you think about it, this has to be a pretty gross experiment. I don't know about you, but I have a fear of maggots. Like every now and then, like in the summer, like when I take out the trash, like sometimes, at least once a summer, there will be like maggots in the garbage bags. I don't know how it happens, and I would freak out. My wife is not afraid of it though. So every time I just call her out and she would clean it up. She's like, I don't know why you're so afraid of maggots. It's not like they would jump on you. Look at them, like they just crawl around. I don't know, it just creeps me out. But there we go. Okay, 3 groups here. Okay, so please, someone tell me, what is the difference? Not what is the difference, but what is the independent variable in this case? Independent variable, and what is the dependent variable? Okay, please tell me. Start with the independent. What would the independent variable be in this experiment? That's right. Okay, so the one that he deliberately changed is the type of jars, right? Type of jar. Okay, so by using the different kind of jars, he wants to know whether or not maggots and flies will form. So the dependent variable is the presence of flies and those white things, maggots. Okay. The meat itself is not a variable. Okay, it's not a dependent variable. Okay, the meat is what we would call a standardized variable, something that needs to be kept unchanged in all three groups, right? Okay, because if you use different meats and then some of them have maggots and some of them don't, then you don't know. Maybe some meats gives maggot and some meats don't, right? So we want to keep the meat the same, okay, and we will only want to change one thing at a time. And that thing we're changing is what type of jars we are using. Okay. And then we want to know whether or not maggots will form. Okay. So what were the results of Rady's experiment? Well, if you watched the recording, you might remember, and you know, using common sense, you probably would guess the results as well. The open jars, you would have the flies, right? Flies buzzing around, and then you would have the maggots. Okay, so group one, maggots and flies. Okay, number two, any maggots and flies? Yes, there were also flies and maggots, but they are just crawling on the netting, nothing on the meat itself. Okay, so maggots, I guess we can say maggots plus flies on the meat. On the meat. Okay, over here, maggots plus flies on the net. Okay, must have been a really fine mesh. Okay, so nothing falls through. And then the last one, nothing. Okay, no flies, no maggots. I wouldn't suggest eating that piece of meat though, but there is no maggots, no flies, which is kind of what you would expect, right? Okay. So what is the conclusion? What is the conclusion? Well, it's quite straightforward. The conclusion is meat does not produce flies or maggots, right? Only flies produce flies, okay? Or maggots. Really, they lay eggs and eggs become maggots, right? The fly's not going to give live birth to another fly, right? That's just not how it works. Okay, so that is the conclusion. We good so far? All right, very good. Moving on. What prompted Pasteur to perform his experiment? So there's another guy, right? Another guy, Louis Pasteur. He's the same person who came up with the method of pasteurization. Anybody know what pasteurization is? Anybody heard of pasteurization? What kind of product in your fridge requires pasteurization? That's right, milk, right? Okay, the milk that you buy from the stores, they're pasteurized. You can't just drink the milk straight from the cow. Okay, you're gonna get sick because there's like bacteria in it. Okay, same thing for like what's it called? Honey, right? Honey. Okay, there are pasteurized honey and then non-pasteurized honey. The pasteurized one, basically it means it's sterilized. Okay, they kill all the bacteria in it. Okay, so he's the guy who came up with that process. Okay, so what prompted him to perform his experiments? Well, not everybody was convinced by Rady's experiment. Okay, so not everyone was convinced by Rady's experiment, the fly experiment, that is. Okay. Many believe spontaneous. Spontaneous generation occurs on a microscopic level. Okay, so they're like, it might not be true for big organisms like flies and maggots, but on a microorganism level, right, for the bacteria, for the microbes, that could be still true. So that's what Pasteur is going to try to find out. Okay, so his hypothesis is this: if cells arise from non-living things, then they should appear from sterile broth. Okay, so sterile just means like treated with heat. To kill off everything. Okay. So here is his experiment. Okay. Identify the control group. Okay. So just to refresh your memory, the control group is the group with no treatment, okay, or modification. The experimental group is the group that is receiving the treatment, okay? The group receiving treatment or modification. When I say treatment, I don't mean like necessarily like a cure for a disease or something like that. Treatment just means like you do something to it, right? Will we be tested on our knowledge about Pasteur experiment? Yeah, there could be questions about Pasteur experiment, okay? And I'll give you an example afterwards, okay? All right, so over here, which one is the control group? Is it the bottle with no special neck or the bottle with the swan neck? Which one is the control? Okay. Why don't I do a quick poll to see if you guys are with me? Okay. If you think the left one is the control, please choose A. If you think this is the control, please choose B. Which is the control? All right. Looks like some people think it's this one. Some people think this one. The correct answer is this one. This is the control. Okay. This is the one that, you know, nothing is done to it. They didn't put a neck on it. Okay. And then this will be the experimental group. Experimental group with the swan neck top, right? Okay. The swan neck top right here. Swan neck top. Okay. What was the independent variable in this experiment? Okay. What's the one that was deliberately changed? Well, just like I just told you, right? It will be the type of flask used. Okay, is it like an open flask or is it a flask with a swan neck? Okay. Next one. How did Pasteur ensure the initial condition of all the flasks were identical? Okay, so he used the same broth. Okay, he treated the broth to kill off any pre-existing microorganism, right? Any pre-existing microorganism. Okay. Hang in there, guys. We're gonna have a break as soon as we finish this question. Okay, I know it has been a long hour. What were the results of Pasteur's experiment? So microorganisms were only found in the control flask. Okay, so for those of you who missed this experiment, basically he left the flask out for, I don't know, like maybe a week or so, and then he checked the broth after a week or so, okay, to see if there is microorganism in it. So only the control flask, the one with the straight neck, only that one has microorganism in it, okay, and not the swan neck flask. Okay? I mean, it makes sense, right? If you think about it from what we know now, if it's open like that, then airborne microorganism would fall in, and then they would love the broth, replicate in there. If it's swan neck, the microorganism will fall, and then they would be stuck here. They're not going to be able to crawl up here, so the broth would remain sterile. Okay, so the conclusion is that living things do not come from non-living things. Okay, so he rejected the spontaneous generation hypothesis. Okay. All right, why don't we take a 5-minute break, okay, just to stretch your legs, grab a drink or something, and we'll resume at 9:40. If you have any questions during this time, please feel free to leave it in the chat, and I'll address it when we come back, okay? So 9:40, we'll come back at 9:40. All right, and we are back for more bio. Why don't we start with another poll question? Okay. Go ahead. Okay, just a couple more minutes for people to try. Sorry, a couple more seconds for people to log in their answer. Sarah, could you clarify your question? What do you mean, which pages of the notebook to read?
So do you like put like a downloadable textbook on our home screen?
Oh,
And
yes.
you said that I think it was like the last time we had a lecture, you were like, oh, you all like the information that you need is basically just in your pre-recorded like lessons and stuff. So I wasn't sure if you were just putting the notebook there for like extra
Yeah, so the text,
info.
right. The textbook is just for extra info. And if you want to know which part of the textbook to read, if you need to read, you can go, you can go back to the lecture folder. Okay, lecture folder. And each of the folder would have a link to the specific section in that textbook.
Okay.
Okay, so rather than downloading the whole thing and look for it yourself, you can just click on the link, right? So for lecture 1, it will be this part and then so on and so forth. Okay.
Oh, okay. Okay, great. Thank you.
No problem. Where am I? Okay, over here. All right. That was 3 minutes. Some of you are not answering. Not sure if you are not sure about the answer or you not there. But do try to participate if you can. So the correct answer is A. Okay, so we're looking for the false statement. So this statement is false because the presence and absence of maggots and flies, as we discussed before the break, that should be the dependent variable, not the independent. Okay, the independent is the type of jars that was being used, right? Okay, so that is the correct answer. Correct in the sense that it is the false statement. Okay, let's quickly do another one. Just gonna do 1 minute for this one. It's quite short. I should tell you, some of these questions are directly taken from the quiz pool, right? So, you might want to rewatch this recording later on. Or like if anytime there is like a poll question or something, like maybe you want to snap a picture of it or something like that, okay? Yeah, majority of the people got it right. The correct answer is the type of flask used. By the way, while we're on the topic of quiz and tests and whatnot, the quiz will be different for everybody, okay? Because the questions are taken from a big pool of questions. So I have a pool, with 50 questions or 60 questions or whatever, and then the platform, the eCentennial, when it generates the quiz, it will randomly pull questions from that pool. Okay, so just to let you know, it is not going to be identical to another person. So if you were just talking about with another friend and you're like, oh, I didn't get that question, that's totally normal. Okay, moving on. Where are we now? Okay, so long answer questions. Let's do the first long answer question here, and then actually, you know what? Should we do the first one? Yeah, why not? Let's do the first one. Okay, let me copy it to a new page so we have more room. Okay, so right here. You know what? I'm gonna actually do it at the very bottom because I'm gonna post this online afterwards in case you wanna, revisit some of the stuff that I wrote down. So here it is. Okay, you are the lead scientist in the study that sorry, in a study to determine if taking a multivitamin supplement reduces the risk of catching a cold. Okay, so let's write a hypothesis for that. Okay, anybody want to give me a hypothesis? Maybe unmute if you're comfortable. Probably is faster that way than typing it out. Or you can type it out. Okay, whichever one you prefer. Okay. Michael asks, for the tests and quizzes, will it tell us the correct answers at the end? Okay, so that is a good question. I forgot to mention. No, it will not. Okay. In fact, and you guys probably will not like this, but we, not just me, okay, but like we, as in like all the science teachers, we have decided that we will not be returning the quiz or the test to the students. As you understand, this is online learning, right? And even though I have 50 questions, but if everybody starts to get their tests or quizzes back, and you know the question pool gets leaked out, so to speak, and it get posted on the internet or whatnot, then it compromises the integrity of the test bank, right? So for that reason, we will not be returning the quiz. So in other words, you will not know which question you did wrong. I know that is not ideal, and it's not great, not perfect, right, for learning, but we just have to, you know, do it that way. Having said that, I don't know what other teachers will do. What I will do usually is I will go through everybody's quiz or test, and then I will pick out questions that are poorly done by a lot of students. Okay, so like I might be like, oh, this question's like, I don't know, 50% of people got it wrong. And then I will talk about that question during the tutorial with the class. Okay, so you might not see which question you got wrong, but I will go through questions that are commonly done incorrectly, okay, with the whole class. So again, I know it's not ideal, but yeah, that's kind of the way it is. Hope that answers your question, Michael. So over here again, hypothesis for this experiment. Okay, so there is one. After taking multivitamin supplements, the risk of a cold infection will reduce. Yeah, that is an excellent hypothesis. Okay, so basically you will say, taking multivitamin, oh my goodness, vitamin will reduce the risk of a cold infection. Okay, it's also as valid to say taking multivitamin will not reduce the risk of a cold infection. Remember, it's just a tentative answer, right? And you can take either stand. It doesn't really matter. Okay. Part B, what is the independent variable and the dependent variable in this case? So the independent variable is the one that you deliberately change. Okay, so it would be whether or not the person takes vitamin. Okay, so taking or not taking the supplement. Okay, and then the dependent variable is, what's the dependent variable? Anybody? Yeah, catching a cold or not. Catching a cold or not. Okay. What group should be involved in your experiment? Okay, so because we're dealing with humans, and humans are complicated, we need to have three groups. We would have the control group. No supplement. You guys don't take anything. We would have the treated group. Yes, supplement. Take the pills. Take the blue pills. And then what's the third group? That's right, placebo group. What is a placebo group? Anybody want to explain that?
It's like a sugar pill group. So it's like if somebody gets a pill that is told that it's like the supplement, and it's actually not. It's actually just like a complete like default pill. It does absolutely nothing. To
Excellent.
see if like people react differently, like psychologically, maybe they think like they're getting the treatment and they're not. I don't know.
That's perfect, right? That is exactly what a placebo is. Sham treatment. Okay, a substance that has no known effect. That's right. Okay, so if we're trying to study the effects of the Pfizer vaccine, you would have the control group, no vaccine, treated group, Pfizer vaccine, placebo group, a vaccine that has no active ingredient in it. Okay, so those would be the three groups. And the purpose of that is to eliminate psychological effects. Very good. The next question, what is the next question? What factor must be kept constant between the various groups in your experiment? So these are what we call standardized, okay? Standardized. Write this down, okay? Standardized variables, because in the lab, I will ask you about this. The purpose of standardized variable is to ensure consistency between the various groups, okay? So that if we observe any difference between the groups, we know it was due to the independent variable and not anything else. Let me say that one more time slowly. Standardized variables are variables that must be kept the same in all the groups. And the purpose of it is so that when we observe a difference between the groups, we know that difference is caused by the independent variable and not anything else. Okay, so what I want to do with you right now is, let's try this. You guys seem to be a very engaging group, and I love that. I'm going to post a link in the chat group. Okay, you're going to go ahead and click on that, and it will take you to a web page. Okay, so go ahead and do that. I will share my screen one. Okay, if you click on the link, it will take you to this page, and then it says list 3 standardized variables. So I want you guys to type in 3 standardized variable for the experiment that we are doing. Okay, the multivitamin experiment. Okay, go ahead and try that, please. Here is a QR code if you are not able to click on the link for some reason. Okay, so I'll give you guys maybe a minute or so to answer. Again, the question is list 3 standardized variables that we must have between the control, treated, and the placebo group in this experiment. And the experiment being whether multivitamin supplements will reduce the risk of catching a cold. Okay, I have 10 people who have responded. Just going to give another minute or so. Please do participate. And obviously, if you're watching this on YouTube later on, you won't be able to do this question, right? But that's okay. You can find out what the results are, which is equally exciting. Okay, looking pretty good. Let me see. Okay, so more answers are still coming in, but let's take a look at some of these. Okay, if you're still doing it, no problem. Feel free to keep typing the answers. Okay, so again, just quick recap. The question is, we got 3 groups here: people who are not taking the pill, people who are taking the pill, and people who are taking the fake pills. We need to make sure everything else is as consistent as possible, which is what the standardized variables are. Okay, so I don't know if you guys know how to read these things, but basically the bigger the word, the font size, that means like a lot of people mentioned that thing. Okay, so a lot of you mentioned age. That is correct, right? You don't want to have like all the old people in one group and all the young people in another group, right? Because that wouldn't be a fair gauge in terms of their immune response, right? Their immunity in general. So you want to have similar age group in all 3 of the groups, okay? Which is why, you know, with the Pfizer data or like Moderna data, right? They test the vaccine in different age group, right? They don't test it in people above 70 the same time that they test it in people between 30 and 50, right? Because of this concept of standardized variable. Okay, diet is another big one. Yeah, you want them to be like eating similar food. Okay, you don't want the vegans to be in one group and then the meat lovers in the other group. Right? You want to mix it up a little bit, have equal representation. It's impossible to completely control exactly what they eat, but you want it to be fairly consistent, right, between the different groups. Okay, gender, yeah, equal ratio of gender in each of the group, definitely. Overall health condition, excellent. Same. Now, here are some of the other ones that I'm gonna mention. Okay, so same vitamin brand. Remember, and this is very important. Standardized variable is referring to things that you keep consistent in all 3 groups. So same vitamin brand would not be a standardized variable because who's taking the vitamin? Only the treated group, right? The placebo and the control group, they're not taking vitamins. So it's not possible for me to make sure that they have the same vitamin brand, right? And for that reason, it is not a standardized variable. Does that make sense? Let's see what else. Okay, so frequent hand wash. So you want to make sure that they have the same hygiene practice, right? That they, are doing similar things to protect themselves from potentially getting a cold, right? So, for example, like if people are wearing a mask to go to work, you want all 3 groups to be doing the same thing. Let's see if there are other ones. Same temperature. Yeah, like if they're in the same geographic location, probably that would be okay. Now, placebo is not a standardized variable, right? Placebo is the group, right? The group that's receiving like a sham treatment, okay? But yeah, most of these other things seem reasonable. Patient, not sure what that means. A cold, that's not a standardized variable, right? Whether they catch a cold or not, that is the dependent variable, okay? Time pills given. Again, that could only apply to one group and not the other. Therefore, it's not a standardized variable. Is that okay? Anybody need any clarification or we are good? Ethnicity, that's another good one. Yeah. Okay, so I hope that makes sense. And if you're still not sure, feel free to send me an email, and we can definitely talk more about it. Okay, what result would support your hypothesis? I'm just going to do this verbally because we're running out of time. If people who are taking vitamins, they have less people getting sick from that group, then you know the vitamin works. Okay, if it's all the same, right? Like maybe 50% of all the people get sick in all the groups, then you know the vitamin is not really helping. Okay, what result would refute your hypothesis? Yeah, exactly what I just said. Okay, if there's no difference between the groups, or like the people who are taking the vitamin actually do worse. Okay, that's possible sometimes in some experiments. And that would refute. Refute means you would reject, right? You will reject your hypothesis. Okay, that is that. Let's move on to the next one. A farmer wants to determine if feeding cows with high protein diet would increase milk yield. Okay, so hypothesis. I'm just going to do this verbally again. It's quite straightforward. Okay, you either think it will or it will not. And remember, do not ask a question in hypothesis. You're trying to answer a question. You don't answer a question with a question, right? Okay, unless you're a teacher, then you always do that. So the hypothesis is feeding cows with high protein diet will increase milk yield. Okay, or you can say will not increase milk yield. Pick one. Doesn't really matter. Okay, so how are we going to set up the experiment? Okay, so we're gonna have cow, one group of cows, okay, and we'll call them the control group. Control group. Okay, what should we feed the control group? Anybody? What are we gonna feed the control group? And then we're gonna feed the treated group as well. Okay. So what should we feed the control group? Okay, so Tyler said we should treat we should give the control group standard diet, and then Tina say high diet. What are we gonna do? Well, for the control group, we will be giving normal diet. Okay, normal diet. We're not gonna like take out proteins. We're just gonna give them like normal things, whatever you give them normally. Remember, control group is the group that you don't do anything special to it, right? You don't treat them, you don't manipulate them, right? At all. No changes to the control group. So that's just normal diet. And then the treated group would be high protein diet. I don't know what you feed cows, okay? I don't know how you can give them high proteins, but you're gonna do that, okay, for this group. And then you're gonna check to see if their milk yield increase, right? Okay, before and after the diet. Part C, is a placebo needed in this experiment? Why or why not? Should we include a placebo group? Should we have a third group of cows where we tell the cows, I'm gonna give you high proteins diet, okay? But we're actually not really gonna do that. It's gonna be like a sham diet. Should we do that? Okay, so a couple of people say no. Yeah, no, we don't need a placebo for cows. Okay, cows don't lie. Okay, they're not gonna be like, I'm gonna hold out on my milk. I'm not gonna produce as much because I know you're doing experiments on me, right? Like placebo is only to account for psychological effects when we're dealing with human subjects. Okay, so no need, okay, because no bias from cows. Okay, yeah, they don't have the brain to know what is going on. Yeah, that's pretty much it. Okay, they don't know what's going on, right? They're not gonna have any bias. Wonderful. That is that. Okay, so I want you to think about standardized variable, right? As well. Okay, I know the question is not asking it. Okay, there's another question from Ro. Do we need a placebo group when we do? Yeah, for humans, we usually include a placebo group, okay, to account for bias and psychological effects. Okay, standardized variable. Okay, so what kind of standardized variables? I'm just going to list a few. You want them to be in the same environment, right? You want the feeding time to be the same. Okay, you want the feeding material to be the same except for the protein content. Okay, you want the cows to be similar age. Okay, gender is not a problem. Okay, usually when I put this question on in a test, when people used to write tests in class, a lot of people put gender, but gender is not a thing. Okay, for this question because only female cows can produce milk, right? Okay. You're not gonna have like male cows in this experiment. Okay. Ananya asks, do we need placebo for kids? Yeah, you'd be surprised. Okay, kids, they do have bias. Like, I mean, my younger daughter, the 4-year-old, you know, she might fall and hurt her knees, and then, you know, I would give her like a little towel and put on the wound, and then I was like, this will make you feel better, and she's like, oh yeah, it feels better, right? But in fact, it's not doing anything, right? So kids do have psychological effects. But sometimes the word placebo is a little bit misleading, right? Because it's not completely for the subject, right? For the experimental group, right? It's also for the researcher, right? You want to include a placebo so that the bias from the researcher is also going to be eliminated as well. Okay, next one. Describe the main principles of, sorry, it's missing a word here, of experimental design. Okay, so I'm going to list some here on the side. In other words, right, what do you need to consider in an experiment to make sure that it is credible, okay, that the result that you get are believable. Okay, so first of all, you have to have proper groups. Okay, sorry, it's a little bit crowded here. But I believe I post a Word document, so feel free to, like, you know, hit a few enter before you print it. Okay, so have proper groups. Okay, you need to have the treated, control, and placebo wherever is needed. Okay, you want to consider things like sample size. Okay, you don't want to just test it on, like, you know, 5 cows. Okay, I got 5 cows with normal diet, 5 cows with high diet proteins, and I'm going to check the milk. Okay, that is a small sample size. Okay, and as you know, with small sample size, the results might not be accurate. Which is why, like, if you think about the pandemic, right, the vaccines, right, when they were doing the testing phase, we hear numbers like, oh, the Pfizer vaccine is like 99.7% protective or whatever, and then when it comes to the real world, that efficacy has decreased to, like, you know, 70% or 80%. So what happened, right? Well, basically, in the trial phase, you were dealing with, like, you know, thousands of people, but in the real world, we're dealing with like millions of people, right? So as the sample size increases, the data is going to be more accurate, right? It's more accurate representation of what it actually is going on. So when we do an experiment, you want to include as big of a sample size as possible. Any other things we should consider? Any suggestions? Looking for like maybe 1 or 2 more. Anyone? No, that's okay. So you want it to be randomized, right? Randomized, like when we do set up the different groups, you want the groups to be representative of, you know, ethnicity, age, that kind of stuff. You want it to be randomized, unless the purpose is to do the experiment in a target group. Okay. And another thing is, you want it to be a double-blind experiment. Okay. So the difference between a blind and a double-blind experiment is that in a blind experiment, the people who are participating in the experiment do not know whether they're receiving the placebo or the treatment. Okay. In a double-blind experiment, even the experimenter, the researchers, they don't know. Okay, they don't know which group is given what. Okay, and then there is usually like a reveal a meeting, okay, where like the third party holds the key to the study, right? They are the people who know who gets what, and then they will review the results. And the third party is usually not invested in the product, so they're not going to give like a bias, assessment. Okay, explain the importance of placebo. We did that already. We're not going to do it again, and I just explained this one. All right, let's see if there's other poll questions. This is just one about hypothesis. This is one about control group. Okay, let's do this one. This would be an example of application question. Okay, you will have to think about it a little bit. Okay, just maybe another minute or so for people who are still deciding. Okay, let's end the poll, show results. Yeah, most of you got it right, which is very good. Okay, so here we have 1 ml of some kind of drug diluted in saline, and you inject it into pregnant mice. So we're not gonna use male mice because we're using female mice here for the treated group. So we should use female mice for the control as well. And they're pregnant. So we're not going to use non-pregnant. Okay. So the treatment group is the one that is receiving the drug, and therefore the control group is the one that is not receiving the drug. So the answer is E. Okay. Pretty straightforward, I hope. All right. Moving on. The graph below. So these are some examples of application questions, scenarios that you have to think about. So the graph below shows the effect of seeing lozenges in reducing cold symptoms duration. Identify additional information that are needed to improve the credibility of the results. Okay, so let's say I'm a salesperson, show up at your door, and for some reason you open the door, and I say, hey, I got this lozenge, and it's shown to effectively reduce the number of days that you have cold symptoms. And here is a graph to prove it. Okay, what kind of questions are you going to ask me to increase the credibility of the results? First of all, you should never buy anything from a person who shows up at your door. But just saying, what kind of information, additional information we need to get? That's right, the size of the group, right? There's no information about how many people were in the studies, or, as far as I'm concerned, it could be just like, me and myself. Okay, so that's not enough. Okay, so you need to have sample size. How many people were involved? Okay, what other questions should we consider? What other information would we required? Anyone else? Sure, age group. Anything else? Yeah, was the study randomized? Was it, so basically like all the stuff that we talked about in the previous question, right? Was it randomized? Was it double blind, right? I have a few other key ones that I want to see. Anyone else? Original health condition, yeah. Good. Still have the key ones that I'm looking for. Anyone else? Dose size. Yeah, how many lozenges do I have to take? Do I have to take the whole pack? Yeah, could be. Okay, so another one is who funded the research, right? Who funded it? Okay. I'd be concerned if the research is funded by the same company who makes the lozenges, right? Okay. In the lecture, I talk about code FX, right? Okay, so you guys, if you don't know what that is, you can look it up. Search it on YouTube and then search up like Marketplace. That's like a CBC show. Okay. So search these two things, watch it over lunch or something. Highly entertaining. Okay. It's related to what we are talking about. Because basically they funded their own research, right? And tells people that the stuff worked. Another one, yeah, side effects, right? Side effects could be. Okay. Okay. And another thing I was looking for is this thing right here, right? Mean number of days until cold symptoms are gone. So, you know, how are these measured? Okay, like, do you have a checklist, or like are people just saying that they feel better, right? Okay, it could be a very subjective thing, right? Okay, so do you have like a standardized list that you check off, right? Like a scale, perhaps, right? For people to use. Okay. Yeah, number of days. Good. All these things. Wonderful. Next one. Here is a new drug called Sleep. Is being tested for its effectiveness in reducing symptoms of jet lag. Okay. For those of you who don't know, jet lag is when you travel to a different country with a different time zone and you can't sleep at night. Okay. A double-blind experiment was concluded. The graph below shows the experimental data from the study. Based on the experimental results, what conclusion can be made about the effectiveness of the pill? Okay. Does it work? Does it not work? Let's do a quick poll. If you think it's effective, please choose A. Effective. B, not effective. No effects. C, not sure. Like, as in like you are not sure. I think I talked about this in the lecture as well. Okay, good. And many people are saying it is not effective. Okay, it is not effective. That is correct. Okay, so let's take a look at why. Right? So if you look at the control group is the group who is not taking the pills, and you can see like 70% of it, 70% of the people who are in the control group says that they having jet lag. Okay, so a lot of people have jet lag if they don't take the pill, which is what you would expect. Okay, and then you look at the pill, and people who took the pill, right? Only 40% of this group report a jet lag. Okay, so if you just look at this and this, right? If we just ignore the middle one for a second, we just ignore that middle guy. I'm gonna take that away. If we ignore the placebo, if I just show you this, you'd be like, oh, looks like this works because the pill, right, caused less people to have jet lag, okay, compared to control. Right? So at first glance, it might look like it's working. However, we must consider the placebo. The placebo are the people who are receiving a sham pill. So you tell them, oh, take this, it will help you sleep, okay? And then they take it. And then you look at the group, and also about 40% reported less symptoms. So in other words, you can give them the actual thing, or you can give them something that's fake. Both will achieve the same result, which tells you it is not the pill that's helping people not get jet lag. It's just the fact that you're telling them here is something that would help with the jet lag. Okay, so if the placebo and the treatment group performs similarly, then we know the drug does not work. Okay, so let me say that one more time. The key thing to know is that, I'll write down, if the results from placebo and treatment group results are similar, then the pill does not work. Okay, so for it to work, the graph would look something like this. Okay. If placebo and control are significantly higher than the sleeping pill here, then we know it's effective. Any question? Everybody happy? Right. Last page. Fill in the blanks. What's going on here? Something is the ability to maintain stable internal environment despite of environmental changes. That would be homeostasis. Okay. Can be physiological or behavioral response. Something is part of discovery science where scientists make generalization from many small observations. Okay, so you're going from specific to generalization. That would be induction. Whereas deduction is logical reasoning used to make predictions about specific case based on generalization. A hypothesis is a tentative, testable explanation for an observation, for a question. And in order for the hypothesis to be valid, it should be testable and falsifiable. Okay. Falsifiable. Oh my goodness. There we go. The variable that is measured or observed, that is the dependent variable, the one that you are not deliberately changing. And the independent is the one that you are deliberately changing or manipulating. A sham treatment is called a placebo. Very good, Marlon. And a study one where subjects do not know if they're receiving a treatment or placebo, that is a blind study. Blind. They don't know. They cannot see. And then this is the double blind. Double blind. The subjects don't know, and the researchers don't know either. Someone else knows, some third party. Relationship between two variables, that is a correlation. Remember, correlation does not mean causation. Just because two things are related doesn't necessarily mean one caused the other. A primary source is a researcher's original report. Books, news report, and magazines are examples of secondary sources. And that's it. Okay, so thank you all for coming. Please continue to come. It's very important that you do attend these sessions. I will post a recording of this online afterwards. I will also post this like filled in versions of the study guide online as well. I will create a new folder, okay, on eCentennial that says study guide answer or something. And you'll be able to find a collection of them as we go on in the course. Okay. All right then. No, remember the next class is next Wednesday. Okay, we do not meet on Monday. Monday is for asynchronous learning only. All right. Thanks again. Have a fantastic day. Bye for now.